Okay, so let's get back to microbiology now. Yeah, you too. Microbiology. Okay, so right, so now we're going to look about bacterial growth, how to measure it, the different phases of the bacterial growth curve, and actually, I also let me just stop this right now because I would like to be able to see my notes here. Go back again. Oh yeah, okay. So how to measure bacterial growth, different phases of the bacterial growth curve, and then the environmental factors that can impact on bacterial growth. So we have these things to do in about an hour and a half, a little bit less. Okay, so there are, well, bacterial growth. If you want to know whether your bacteria are growing, you have to be able to measure them. And when I say bacterial growth, I'm not talking about one cell getting bigger. We're talking about bacteria becoming more numerous. So you have to have a technique to count them. And one of the big classical ways to count the number of bacteria you have in a sample is to start off making a dilution series with 10-fold dilutions, and then you'll take a little bit from each of these 10-fold dilutions, spread them out on a plate, put them in the incubator, and then count the number of colonies that grow. So each colony comes from one live viable bacterium that was present in the sample. So you count how many colonies you've got, you multiply it by the dilution factor, and that's the number of bacteria that you have per ml in your original sample. So that's what you'll be doing in the lab classes. So that's okay, works, and it gives you, the unit here is CFU per ml, colony forming units per ml. So whenever you see this, then you should be able to visualize the technique that was used to make the measurement. Now the other thing that you can do to measure, yeah.
Excuse me, I read the lab first station, and I was wondering, inside the tube, does the liquid is the agar for the dilution? In which solution are we treating the bacteria?
Okay, so this is a question about the lab classes.
Yeah.
I think you just dilute them in sterile water, but we have one TD class next week, okay, and we'll be going through the lab class protocol next week. Okay. Yeah, so CFU per ml, it gives you a measurement of the number of live viable bacteria in a sample. So the other way to measure how many bacteria you have is to take a turbidity measurement. So if you've got some kind of liquid and you shoot some light through it at a wavelength of 600 nanometers, so mostly if you've got a clear solution, then all the light will be transmitted. If you have some small solid objects in suspension, then, like bacterial cells, crystals, something like that, then these objects will scatter some of the light. And you can measure this as an optical density at 600 nanometers wavelength. Okay, it's not absorption by a chemical, it's just light scattering. Anyway, so your OD 600 measurement will give you an indication of the concentration of particles inside the suspension. So we're talking about bacteria. The OD600 will be directly proportional to the number of bacteria per ml. However, live and dead bacteria will still scatter light here. Okay, so the relationship between OD600 and number of CFU per ml is more or less 1 OD600 is about 10 to the 9 CFU per ml. Okay, but it varies between bacterial species because bacteria have different sizes and different shapes. So depending on that, the specific relationship between OD600 and the number of bacteria will be different. Okay, so it's 1 times 10 to the 9 CFU per ml plus or minus a factor of 10 depending on the bacterial species. Okay. Now, if you want to measure bacterial growth in liquid medium, the kind of experiment you perform is you get your bacteria, you put them into LB medium, complex medium, and then take samples at different time points and then measure the number of live viable bacteria as CFU per ml, and then plot the number of live bacteria as a function of time. And in very many cases, you'll get a curve like this. So at the beginning, then you'll have a latent phase. So at this time point, bacteria don't start growing immediately. Now, why does that happen? Well, often when you start a culture, you'll be taking bacteria from an agar plate or from an old culture that might have been lying around in the fridge, conserved at 4 degrees for a certain amount of time. So those bacteria will have shut down their metabolism. And then when you put them into new, fresh medium, very rich in all sorts of nutrients, then they'll start to activate their metabolism, start expressing transporter systems for importing glucose, the enzymes required to metabolize glucose, this kind of stuff. So this takes a little bit of time for bacteria to adapt to the new conditions. So that's what's happening here. Once they do adapt, then they move into the exponential phase of growth. So during the exponential phase, they are growing at a constant rate. That means the number of bacteria will double at a constant rate every hour or every 2 hours or every 30 minutes or something like that. So that's why when you plot the number of bacteria per ml on a log scale, okay, so this is a log scale here, not a linear scale, then you've got something that gives you a straight line on a logarithmic scale. So that's an exponential curve here, with bacteria doubling at a constant rate. So they're happy, they're just going like the clappers, growing like crazy. But after a while, they will either begin to run out of food, they'll use up all the peptones or all the glucose in the medium, or they will produce too many toxic products. So bacteria will tend to ferment glucose or other sugars and acidify the medium, and well, most bacteria don't like to grow in acidic medium. So this in acidic conditions. So this will lead to the stationary phase where bacteria stop growing. So the conditions for growth are not so good anymore. The medium is too acidic, they've run out of nutrients, something like that. Now, bacteria don't die immediately under those conditions. They start expressing a certain number of starvation proteins. So these starvation proteins, if I just read my crib sheet here, what do they do? Yeah. So their general effect is to make the bacteria more resistant to stress. So they will stop producing peptidoglycan. They will stop producing autolysids. Ah, that's not right, actually. The cell wall tends to be reinforced, so they'll stop producing autolysids, that break down the cell wall at the same time as peptidoglycan is synthesized so that the bacteria can grow and divide. What they'll tend to do in the stationary phase is stop the activity of autolysins so the peptidoglycan doesn't get broken down, but keep synthesizing peptidoglycan so the cell wall becomes thicker and more stable. Also, they'll tend to produce chaperone proteins which can prevent other proteins in the cell from denaturing. And so generally speaking, they become more resistant to chemical stress, to heat stress, and to antibiotics. They're a bit tougher. So there's a kind of like a survival mechanism. But this only works for so long because all of these bacteria here are still metabolically active and they still need some kind of organic carbon source to run their metabolism. And when this runs out, well, that's it. Then they start to die. Okay, then you get to the death phase of this curve. Now, it's not really obvious what death means for bacteria. Okay, and one of the concepts was not really that new anymore, but certainly new compared to when I was learning microbiology as an undergraduate. And one of the relatively recent concepts is that during what's called the death phase, many of the bacteria in the culture are not actually dying, but they are transitioning to what's called a viable but non-culturable state. So they're still alive, but you can't grow them in standard culture conditions. So that means that when you perform the experiment to grow up to all of this is based on the number of CFU per ml. So you're growing the bacteria up on the agar plate and counting the colonies. That means that the number of culturable bacteria goes down here during the death phase. But are they all dead? Well, many of them are dead, but some of them seem to be cells that are still intact. So they haven't like exploded or anything, they haven't lysed, and they seem to have some basic metabolic activity that remains in these VBNC cells. And so it seems to be some kind of a function that is a state of these cells, which is similar to what you have with bacterial endospores. It's a latent phase or a latent state of the bacterial cell, which has a very, very low level of metabolism, and it occurs because it allows bacteria to become reactivated under some specific conditions, but not under normal conditions of culture in the lab. Now, this is partly a philosophical question of what is it like to be a dead bacterium? That's kind of, I guess it's beyond human understanding, that question. So that's the philosophical part, but the practical part about this is that in the food industry, detecting bacterial contamination, the reference technique is always based on culture, on the differential selective media that we saw just before. And the question is, if you've got VBNC bacteria in there, you can't detect them with those techniques, but they may still represent some kind of danger for people who eat this stuff. Okay, so there's a bit of a discussion about how can you quantify these VBNC bacteria? Actually, are they dangerous? Who cares? Probably they're not going to contaminate anyone anyway. So that's something kind of new about bacteria. So for Thursday, I'd kind of like everybody to think about this a little bit. How can you tell the difference between VBNC and dead bacteria? And we'll start on Thursday with a little bit of a discussion about this. And what I would like you to do, find out what kind of experiments can be performed, how do they work, how do you interpret them. Okay. Everybody okay with that? And there won't be a microbe of the week this evening, so Thursday. Okay, so yes.
We use the technical control line. We could be able to identify some acid amine, right? So if we identify some acid amine, maybe we could know if they still have some metabolic activity.
Okay, Thursday. Talk about this on Thursday. Maybe yes, maybe no, but talk about this on Thursday. Okay, so one of the things that is very useful to determine for very many bacteria is their growth rate and their doubling time. So to determine the doubling time of bacteria, you need to analyze data that is in the exponential phase here. And you can only really see it when you plot the data on a log curve, on a log scale. So these are the same numbers here, the same numbers plotted on a linear scale and a log scale. And with this curve, you can't really, it's very difficult to see where your growth phase, the exponential phase, really starts. Where it's pretty easy to see by eye when you plot the same values on a log scale. Okay. So once you're in the growth phase during the exponential phase, then you can try and determine what is the doubling time of the bacteria, the time required to double the number of bacteria. So how do you do this? Well, you do this with a little bit of mathematics, because basically if you say that there's a constant doubling rate, then that means that over a specific time during the exponential phase, the number of bacteria that will be present after that time t will be equal to the number of bacteria you had at the beginning, your time 0, multiplied by 2 to the power of n, where n is the number of generations. So that means like n is 1 generation, twice as many bacteria. N is 2 generations, 4 times as many bacteria. Okay? And when I say n0, I'm not talking about 0 here, because this is not in the exponential phase. You've got to choose n0 here, and nt is one of these points up there. Okay? So that's your basic equation. What you want to do, you know what t is, okay, because you've done the experiment, you know when you took each sample, so you know this value. What you want to do, what you want to find is to find n here, okay? So you've got to rearrange this and find n. Now, if you've got powers, exponents and stuff like this, it's always a good idea to take logarithms. So you take the logarithm of everything, something multiplied by something else. When you take logs, it becomes addition. Everybody remembers that, right? Yeah. And then when you have exponents, when you have powers of something within a log, then that becomes n times that logarithm. So this turns into this, okay? So that's the whole point of taking the logarithm, so that you can get an expression that's rather simpler, and you just reorganize that, and that gives you the formula to tell you the value of n, the number of generations that have occurred over the time that you're looking at. So you know what t is, you know the time you took these different measurements, you've calculated what n is, and that will allow you to calculate the doubling time, the generation time, okay? So that's the time over which you made the measurements divided by the value of n that you worked out. So that is something very, very useful to determine for different bacteria. Now, I've gone to the trouble of finding out the maximum or the shortest doubling time observed for different types of bacteria. So generally speaking, in the laboratory, for lab class and this kind of stuff, we'll be using bacteria that grow very quickly. That means they have a very short doubling time, okay? So 0.35, this is a bit more than 20 minutes. This is like 0.5, this is about 30 minutes. So E. coli under optimal conditions can double every 20 minutes. Staphylococcus aureus every 30 minutes, 0.5 hours. Now that's not true of every bacterium. So Mycobacterium tuberculosis only doubles every 12 hours. And this has very important consequences when you want to isolate different types of bacteria. So if you want to isolate Staphylococcus aureus from a sample, you can spread it out on a selective or differential medium. And because it has a short doubling time, a fast growth rate, you'll get nice colonies after one night, okay? One day, next day, you'll have colonies. Mycobacterium tuberculosis, well, you have to wait 2 or 3 weeks before the colonies grow because the growth rate is very slow. So that's true for some important pathogens, and it's also generally true for these bacteria down here, which are autotrophs, okay? Anabina, it's a Cyanobacterium nitrosomonas is the example of a chemolitho autotroph. So these bacteria, they have to make everything from scratch, from very, very simple components. And that's much more difficult in terms of biochemistry than what E. coli and Staphylococcus aureus are doing, which is, starting with glucose peptones, stick them together in a slightly different way, and okay, that's it, you've made your cell. The biochemistry that these autotrophs have to perform is very, very much more difficult, and it takes longer for them to get enough energy and make enough chemical components to produce a new cell. Okay, so soil bacteria, autotrophs generally grow slowly. So if you want to find new bacteria with interesting characteristics in environmental samples, sometimes you have to culture them for a long time. So that's what we saw last week, right, with the Loki archaeota. They had extremely slow growth rates. Okay, so that's doubling time and growth rate. Just to make sure everybody has understood this. Yeah, I've got another question here for you guys about that. See if you can get this one. Try to be on purpose, not obvious here. That's pretty good. Okay, pretty good. Right, so here's what I got so far. Okay, so 30 votes. 4 is the wrong answer. Okay, nobody fell for this one. Doesn't matter what bacterium is. It could be anything. It will still be the result will still be valid. 3, well, maybe good, nice try, but in fact, no. There is something you actually need you can perform the calculation just with this information, but will it be accurate? That's the question. Okay, you can perform the calculation. You've got the information you need to perform the calculation, but how do you know it's right? So we had, most people said this one, that's the right answer. 2 is the correct answer. And 1 is not the right answer. So why is that? Why doesn't it matter? Well, if you can think about, this in mathematical terms, okay? So these numbers here, if you just got the OD600, there's a direct proportionality between the OD600 and the number of bacteria per ml. So you'll have some constant K that can relate the two together. So your OD600, so this will be K times the OD600 in both terms here. But you don't really care because that's going to divide out later on, because you're going to be taking K away from both of these. So whatever this constant is that relates the OD600 to the number of cells per ml doesn't matter because it's going to disappear in the calculation later on. So you don't need to know the relationship between OD600 and the number of bacteria per ml. Actually, that's kind of, I put this one in there because in the lab classes, you have to perform this calculation. And some students, they calculate the number per ml and then they run the log on those numbers and they're just making things more complicated for themselves. You don't need to do it. Anybody can give me a quick estimation of, okay, so yeah, so the last thing is 2 is the correct answer because, you know, if it goes from 0,1 here to 0,5 here, you're not getting a correct estimation of the growth rate. You have to know that you're in the exponential phase. So that's why 3 is the correct answer. Similarly, if it goes from 0,1 here and then it goes into the stationary phase, then your calculation will be incorrect. So yeah, good. Well, most people spotted that. So it's going to be about what, roughly speaking, the doubling time here? Don't have to work it out accurately, but just roughly. Nobody? How many doublings have occurred? How many generations? Well, a bit more than 2, right? 0,1 to 0,2, 0,2 to 0,4. That's like 2 generations. The number is 0,5. 90 minutes, so the doubling time will be about 40 minutes or something like that, all right? Also, if you have to make the calculation, always check if your answer is vaguely reasonable. You know, this number is never going to be like 5,000 hours, okay? If you get an answer like that, it's going to be wrong, right? Okay, so all of this stuff that we've been talking about, the bacterial growth curve and stuff like that, that is true for what's called a batch culture. You've got your tube or your beaker or your conical flask full of medium, you put the bacteria in, you grow them over a certain period of time. So that's a batch culture and you get this kind of curve. So another thing that can also be useful sometimes is to grow bacteria in continuous culture. So in that case, what you're trying to do is to keep your bacteria growing exponentially all the time. And the way you can do that is to carry on adding extra medium and you take off excess bacteria continuously. So you need to set up some kind of equipment like this, which is called a chemostat, because the chemical environment in which the bacteria are growing is static. So what is happening here? So this is where your bacteria are growing. You keep this at 37 degrees or 30 degrees, and you stir it around so that the oxygen can circulate. And instead of just like keeping this with a fixed volume of medium, from time to time you're going to get rid of the excess bacteria here, which you collect, and you're going to add in extra medium from the top. And the whole question about this is, okay, how much is the right amount of medium to put in per hour to optimize the productivity? So why would you ever want to do this? Okay, well, in terms of biotechnology, it might be useful for you to run a big lot of production of bacteria in exponential phase, and it might be helpful to optimize your production. And sometimes in research microbiology, it's useful to look at the evolution of bacteria over a large number of generations during which they are kept in the exponential growth phase. So there are some people that do work like this on the evolution of antibiotic resistance or resistance to phage infection in bacteria that are in a chemostat. So the experiment can run over 3, 4, 5, 6 days. And during the whole of this period, the bacteria are kept in the exponential phase. They never run out of media. They never have a difficult, stressful chemical situation. So can simplify the analysis because you're keeping the bacteria at the same growth phase for the whole duration of the experiment. So that's what it's useful for. And so how do you know what the right flow rate is for the new medium that is coming into the incubator? Well, this is the value of D, okay? So D is the dilution rate. And the dilution rate is the flow in terms of liters per hour of the new medium you put in divided by V. And V is the volume of the chemostat. So it could be 0 to, I know, could even be greater than 1. So if you try running a chemostat with different flow rates, on different dilution rates rather, then you can look at the biomass that you get, coming out of the chemostat, and you can look at the concentration of whatever nutrient is limiting. And you get these two kind of curves. What you want to do is to run your chemostat at a flow rate or dilution rate which will keep your biomass production near the maximum. So if it's too low, in fact, the bacteria don't grow very well because there's some limiting nutrient, okay? They've used it all up. So that's why if you've got like a very low dilution rate, you're not running the thing at the optimal, under optimal conditions. However, if your dilution rate becomes too high, it's not the nutrients that are limiting, you'll be washing out too many bacteria. So if we look at the setup again, if the dilution rate is too fast, you'll be getting rid of all these bacteria too quickly, faster than they can replace themselves by growth. So in the end, if your flow rate is too high, then you get much less production of biomass. So the whole point is, okay, you've got to optimize the dilution rate, and you want to work in this kind of range here. Now, in theory, you could perform some small-scale experiments, find out what the optimal dilution rate is, and then set the whole thing up. Okay, I'm going to turn my dial to 0.4, leave it, I'll come back in 4 days' time, everything will be fine. Real life doesn't work like that. So what generally happens is you've got some kind of detector that can control the pump here, which is going to change the dilution rate according to the optical density of the bacteria. So, you set it up and you start at you might say, okay, I'll start at 0.5. But if this turns out to be too high, then the optical density here will start to go down, and then you want to reduce the dilution rate. Okay, so you've got some kind of simple loop to control what the dilution rate is according to the measurement here. Okay, so that's another way to calculate to grow bacteria in a liquid culture. Okay, so now we're going to leave liquids. There are many bacteria that live in a kind of aquatic environment, in the sea or in the lakes and stuff. But a lot of bacteria that are interesting for us actually grow on surfaces. And they don't grow as free bacterial cells floating around in a large volume of liquid. And in these cases, bacteria growing on surfaces tend not to be just, the bacteria on some mineral surface, but they'll secrete a kind of extracellular matrix that they live in. So, and that forms a biofilm. So, biofilms can be monospecies. So, if you get Pseudomonas aeruginosa infection, then these pathogens will secrete biofilm polymer, which is very, very difficult to cough out from your lungs, which is what makes them so dangerous. Staphylococcus aureus, when it contaminates medical implants or catheters, it will grow as a biofilm on the surface. And, one of the things is these bacteria are much more difficult to treat with antibiotics because they are not growing in a kind of liquid medium where a small molecule antibiotic can freely diffuse. They're down in the biofilm where they're kind of protected by this matrix of biological molecules. And so monospecies biofilms, relatively rare. More often, I think, you get multi-species biofilms. So, it's the kind of film that tends to accumulate over time on any surface in your house when you don't clean it very often. Okay, so it's like, the grey stuff that accumulates on your shower curtain. It can be Methylobacterium sphingomosis. Oh, dental plaque, of course, biofilm, with several species that are in there at different levels within the plaque. And okay, so that's like human dwelling, human mouth. Outside of the human environment, you get a lot of biofilms like stromatoliths. So these are communities of photosynthetic microbes that grow on the surfaces of rocks. And if you look, there are several species that are in there all kind of growing together. Okay, so biofilm growth is not an accident. It's a kind of a mode of bacterial growth that is controlled. Okay, let me just get down here. So, and it goes through different. Why is this happening? I never wanted to do. I never wanted to do that. This video is going to be so bad. Okay, finally back to here. Yeah. Okay. So biofilm formation and growth goes through distinct phases. So at the beginning, you'll have free living bacteria in the liquid phase will attach to the surface. So that's phase one. Initially, the attachment is, it's quite weak. Then the bacteria that interact with this surface will express specific adhesins, which could be, for example, pili, to firmly attach to this surface. And they'll grow on the surface just as individual bacteria until they become sufficiently numerous. And after this point, this is phase 3, all the bacteria that are growing locally together will start to synthesize an exopolymer and secrete it. And this will form the the biofilm, bacterial cells inside the exopolymer that they secrete. So for a while, this biofilm will grow. So it's going to get bigger, more bacteria, more polymer, a big chunk of gunge. And then finally, it's going to burst and disperse. And this is also a controlled process. Bacteria will have to express enzymes that degrade the exopolymer and which will allow them to be released into, the surrounding liquid. And of course, this whole thing goes around in a cycle because at the end, you'll release free swimming bacteria and, they're flagellated here. So the expression of the flagellum is going to be regulated. No point in having the flagellum in this environment here. So, different sets of genes will be expressed by the bacteria at different stages in this cycle. Okay. So what do we need to know about that? I better be careful what I'm doing here. Now, one of the important points in this is the transition between stage 2 and stage 3, where you've got bacteria here at stage 2 that have adhered to the surface, but they haven't synthesized the exopolymer yet. This will only happen when there are enough bacteria locally to, make this whole thing work. And that's the next question really, is how do they know, right? How do they know when there's enough of them around there? And this is a phenomenon called quorum sensing. Okay, a quorum is if you're like in a voting or in a parliament or something or an association and a certain percentage of the shareholders or whatever have got to be present or represented, otherwise the vote is not valid. So the idea here is to use this concept and apply it to microbiology, and it was first observed in these symbiotic bacteria in, let me get this right, is it in fish or is it in some kind of octopus? I think it's in kind of, in an octopus. Let me get this. Am I going to check out? Okay, it's not doing anything here. Oh, I didn't write it down. Okay, but they're in these kind of patches. I'm pretty sure it's an octopus, right? Some kind of like octopus. And the bacteria live in patches on the skin of the octopus, and they emit light. And so the people who were looking at this phenomenon, they wanted to try and culture the bacteria and get them to emit light by the expression of these luciferase genes. And they found that they could only start expressing these genes when they were at a very high density, which was the case in vivo, in the animal, that carried them. There were lots of them in these like light-emitting organs. And so that was the beginning of research on how do bacteria regulate the expression of genes depending on how many of them there are. Okay, so this was the first quorum sensing phenomenon that was studied and actually understood. Okay, so what happened in these bacteria is that initially they are expressing a molecule of proteins called LuxI, and LuxI allows the synthesis of a small molecule which is membrane permeable, so it's synthesized in the cytosol and then it diffuses out of the bacterium. So if you have a small number of bacteria, then this molecule here is just going to diffuse away and the concentration will be very, very low outside the bacterium. If you've got a large number of bacteria all synthesizing this signaling molecule, which is called an autoinducer, then the concentration will start to go up. And the bacteria detect the presence of this autoinducer because what it does is it's going to diffuse back into the cell. If the concentration is high enough, it will bind to this molecule called LuxR, and LuxR induces the expression of all the luciferase genes. So they're going to start to emit light. And one of these genes here is LuxI itself. So you get a kind of positive feedback loop. Okay, because the inducer, the autoinducer binds to LuxR, LuxR causes the expression of more LuxI, LuxI produces more of the inducer. So the whole thing is going to, once it starts, it's really going to ramp up very quickly. It's not going to be totally out of control because LuxR will inhibit its own expression. So after a while, once you get like a maximum amount of the autoinducer, there's no more LuxR and the activation of LuxI will stop. So that's the kind of like positive feedback loop. And the critical thing is the concentration of this autoinducer outside the bacteria. So what is it? Well, for Vibrio species, it's N-acyl homoserine lactone. Okay, so what does that mean? So this is a kind of like amino acid backbone here, right? N terminus, C terminus. But this would be serine, except that the hydroxyl group has come along and bound here and made a cyclic structure. So this is homoserine, and it's N-acyl on the amino terminus. And the R group is something, I don't know, acetyl group or methyl group or something else, okay? So many gram negatives use a very similar system for quorum sensing. Okay, so very frequent in gram negative bacteria. Okay, what about gram positives? Okay, so many gram negatives have this system. So in Agrobacterium tumefaciens, this determines when bacteria will start exchanging plasmids, because they detect that there are a lot of them nearby, so they can start producing type 4 conjugation pili, because they have a chance of like attaching to another bacterium. There's no point in doing that if there's nobody else around. In Pseudomonas aeruginosa, very important for virulence. Once there are enough of these bacteria locally somewhere in the lung, they'll start to produce the biofilm, and this will produce a more severe pathology. Okay, for gram positives, a lot of them produce oligopeptides. So for Staphylococcus aureus, this is important for virulence. And in Bacillus subtilis, this is one of the things that leads all the bacteria to sporulate at the same time. So these oligopeptide system, how does this work? So they'll start producing a protein expressed on the ribosome. The protein is going to be cut up into little oligopeptides, which will be exported. It's just like the acyl homoserine lactones. Once the extracellular concentration is high enough, it can be detected by the bacteria. So the detection system is a little bit different. These oligopeptides don't diffuse across the plasma membrane, so they have to bind to a specific receptor, okay? And the receptor is going to signal inside to start to express the target genes, whatever they are. Okay, so that's quorum sensing. Yeah, very important in a lot of like real life situations in microbiology, because just growing bacteria in 200 ml of LB medium doesn't really represent the way a lot of bacteria grow, especially pathogens. Okay, so just to summarize from up to so far, okay, chemotroph, phototroph, organotroph, lithotroph, all this stuff, you have to understand this. Don't ever mix up auxotroph and autotroph. That's well, it's not exactly the worst thing a microbiologist can do, but it's on my list of pet hates, I have to say. I see it in exams when students get this mixed up. Okay, the different types of media, okay, defined, complex, selective, differential. Growth curve, very important to know what the different phases are and how to calculate the generation time. And then for biofilms, what is a biofilm? What are the different stages in biofilm formation and maturation and lysis? How does quorum sensing work? Yeah, roughly speaking, all right? I don't think anybody has to be able to draw the structure of an acyl, N-acyl serine lactone. Okay, so now we have about half an hour, right, to think about how different environmental factors impact on bacterial growth. Yeah, okay, so I think, yeah, I'm gonna, that's why, hey, come back, I didn't want to do that, let's do it again. So I'm going to do them in this order, okay? Temperature first, then oxygen relationships, salinity, activity water, pH, and radiation. Okay. That's funny, why does it do that? Okay, so temperature, right? So if you try and grow bacteria at different temperatures, pretty much for any kind of bacterium, yeah, I think it's absolutely true for every type of bacterium. They're not going to grow equally well at different temperatures, okay? And what you can do if you have plenty of time available and a lot of different incubators going, you could look at the growth curves of bacteria at different temperatures, measure the growth rate as a function of the temperature you use for the culture. So all bacteria will show this kind of bell-shaped curve. So there will be a minimal temperature below which bacteria will not grow. Then after that, if you increase the temperature, you'll find that bacteria will grow more quickly. Basically, all of their enzymes will be adapted to function correctly at a particular temperature. Right? So as you increase the temperature, all of the bacterial metabolism is going to work better. And that will carry on until you reach the optimal growth temperature for this particular bacterium. And then if you carry on increasing the temperature, well, growth rates will decline again because proteins will start to denaturate, membranes will become too fluid and they won't function correctly anymore. And then finally, you know, bacteria will be killed by this extra heat so they don't grow at all. So these are what are called the cardinal temperatures for bacterial growth. Minimal growth temperature, optimal, and the maximum. And you can define these three cardinal temperatures for every type of bacterium. So this is, yeah, that's another question here, right? So I'm talking about growth speed here. What's the valid measurement of growth speed? I don't know if I want to. Can we do this by just a show of hands, or do you want to vote? What do you think? Vote? Oh, everyone's got their phone. Okay. I'll do it. Except. Oh yeah. Oh, come back. Ah, it's gone again. Tell me. Oh, shit. I have to. Yeah, I have to open the question first. That's it, right? Yeah. Oh, it doesn't seem to want to start. Oh yeah, it's gone. Okay, should be working now. Yeah. Yeah, so go ahead, vote. Vote to your heart's content. Okay, let's get about 20 votes here. 20. Oh, that's perfect. Almost perfect here. 23. Okay, so here's what we got so far, 24 votes. And it's kind of split 50-50. So half of you say the doubling time is a measurement of the speed, and half of you say no, it's the inverse. Well, think about it, okay? If your doubling time is 1 hour, are you growing faster than a bacterium with a doubling time of 10 hours? If the doubling time is smaller, the growth is faster, right? So this is the right answer. This is the inverse of the generation time. The longer it takes to double, the slower the growth is. Okay, so that's why I'm like asking this question, because I kind of realize a lot of people might not be clear about this. Okay, so basically that's what this scale is here. Okay, this is going to be 1 over G, the inverse of the generation time at different temperatures. Okay. Okay, so depending on, okay, so the optimal growth temperature will be, the temperature which those bacteria like. Okay, they're adapted to grow at a particular temperature. So depending on what the cardinal temperatures are for bacterial growth, then you microbiologists define these different categories. So perhaps the ones that we'll be working with in the lab are all going to be mesophilic bacteria, so they will have optimal growth temperatures between 20 to 40 degrees. They generally won't grow at all below 15 degrees, and they won't grow above 50. So, mesophiles, if you put your tuna sandwich in the fridge, you won't have to worry about mesophilic bacteria growing on your sandwich, especially in the mayonnaise. However, so Escherichia coli, for example, coli will not grow in the fridge. However, psychotrophic bacteria, well, their growth optimum might be at between 20 and 30, but they can still carry on growing at temperatures down to perhaps 0 degrees. So Listeria monocytogenes, that's the one that can contaminate your tuna mayonnaise, pate, stuff like this in the fridge, because the Listeria can carry on growing even if it's cold. And as well as that, there are some bacteria which are really specialists for growing in cold conditions. Their optimal growth temperature will be 10 degrees. They will not grow at all at like 25 or 30 degrees, and they can carry on growing down to like 0 or even negative temperatures. So at the other end of the spectrum, you have thermophiles, so they can perhaps grow up to 80 degrees, and hyperthermophiles, so these guys will actually prefer to grow at 90 degrees. Some of them can even carry on growing at, more than 100 degrees centigrade. If you put them under pressure, they'll still grow. You don't have to worry about these hyperthermophiles contaminating your experiments in the laboratory, because we're going to sterilize them at 120 degrees, right? But if these guys keep growing, could cause a problem. Well, in fact, no, because they won't grow at temperatures less than 50 or 55. So for a normal experiment in the lab where you're incubating at 30, 37 degrees, hyperthermophiles are just not going to give you any trouble. So in blue here, this is an archaebacterium, and all the others are eubacteria. So how do these, extremophiles manage to grow at these extreme temperatures? So for psychrophiles, their membrane biochemistry is rather specific. They have shorter chain fatty acids, the membranes are more fluid, so they function well at low temperatures. That's one of the reasons why they can't grow at temperatures more than 25 degrees, because at that temperature the membranes become too fluid and are not stable. Okay, so they can perhaps grow in seawater. The salt conditions mean that the cytoplasm of the bacterium doesn't freeze at -2 or -3 degrees. For thermophiles and hyperthermophiles, once again, membrane biochemistry is different, so the fatty acid chains on the lipids in the membrane will be longer and more stable at higher temperature. And there are a lot of differences in protein structure when you compare proteins with the same function in thermophiles, hyperthermophiles, and compare them to mesophilic proteins. So they'll have the internal part of the protein, it tends to be very hydrophobic. So hydrophobic interactions increase in strength with temperature. They have a lot of hydrogen bonds inside the protein, helping to keep them together. And they have a high concentration of proline. So proline, if you remember, is the only cyclic amino acid. It's less flexible than all the others. So if you have more proline, there's less opportunities for the amino acids to, rotate around these two angles. You know about this, right? Okay, well, you will, okay? So the amino acid chain in proteins will be less flexible in hyperthermophiles because they have a lot of proline. Okay, so these are adaptations in thermophiles and hyperthermophiles. Okay, so oxygen relationships. Now, bacteria can have different relationships towards oxygen, going from obligate aerobes, which absolutely require the presence of oxygen in order to grow, to strict anaerobes, for which oxygen is toxic. So in between, okay, so this is supposed to illustrate the results of an experiment where bacteria have been inoculated throughout the tube with a kind of semi-solid agar medium. So obligate aerobes will only grow at the top of this tube where you have a high concentration of oxygen, and strict anaerobes will only grow at the base of the tube where you have very little oxygen. So facultative anaerobes, they grow better in the presence of oxygen, but they can grow anaerobically all the way down to the base of the tube. They do not require oxygen for growth. Aerotolerant anaerobes cannot actually use oxygen in their metabolism, but they can survive it. Okay, oxygen is not toxic to them, so they'll grow equally well all the way up the tube here, but they don't grow any better when oxygen is present. And then you have a kind of rather, I always feel sorry for these guys, the microaerophilic bacteria, because they require oxygen, but too much is toxic for them. Okay, so they will only grow under very specific conditions where, there's some oxygen present, but not too much. Okay. So why is oxygen toxic? Well, oxygen metabolism, using oxygen in respiration as a terminal electron acceptor, can generate reactive oxygen intermediates, hydroxyl radicals, superoxide radicals, which are very reactive and very toxic. So any bacterium that wants to survive wants to survive in the presence of oxygen, has to have some way to get rid of these toxic forms of oxygen. And that's these two enzymes here, catalase and superoxide dismutase. So all of the bacteria that can really grow well in the presence of oxygen have both of these. So superoxide dismutase, what will that do? It transforms the superoxide radical and reduces it to hydrogen peroxide, H2O2, which is also a toxic chemical for bacteria. So you need catalase to break that down into water and molecular oxygen again. So out of these two, it's a superoxide dismutase which is really the most important in being able to survive in the presence of oxygen. So Aerotolerant anaerobes have superoxide dismutase but no catalase. Strict anaerobes do not have either of these enzymes. And microaerophilic bacteria generally have superoxide dismutase, may or may not have catalase. So it's always good to have an idea of some bacteria which represent the different categories. So, Escherichia coli, most enterobacteria are facultative anaerobes. Bacillus subtilis generally thought to be an obligate aerobe, but when I checked this, in fact, some strains of Bacillus subtilis can survive without oxygen. But Mycobacterium tuberculosis is definitely a strict aerobe. Strict anaerobes, Bacteroides and Clostridium, these families, and for microaerophilic bacterium, Campylobacter, apparently. So this kind of leads up to the question of how do you study these strict anaerobes? Because when I was talking about identification of bacteria, all the techniques start with an isolated colony on an agar plate. In your agar plate, you've got, the agar, you put the bacteria on the top and you put that in the incubator. So all the bacteria that grow on there are in direct contact with atmospheric oxygen. So strict anaerobes and microaerophilic bacteria will not grow under standard conditions on an agar plate. So how do you get them to grow? Well, you have to use some kind of setup like this, an anaerobic jar. So what you have here is you've got a setup where you can put your agar plates in here, and you'll have a kind of little sachet of chemicals here, and you add water to this, and then screw the lid down really tight. What you have in here is you have a mixture of sodium borohydride, you have some of that, so that will react with water to give you sodium borate and hydrogen gas. And you also have, this is citric acid and bicarbonate of sodium bicarbonate, and when you add water to that, then they'll react together, produce carbon dioxide and hydrogen. And what you have up in the lid here, you'll have a bit of palladium, and the hydrogen gas will react with all the oxygen that's present in the jar and produce water. So, you produce hydrogen gas chemically, it will react with the oxygen and remove it from the atmosphere. And you can tell whether it's worked because you'll have a strip of paper with methylene blue in it, and in the absence of oxygen that will turn white. So you can put your bacteria in there and anaerobes will be able to grow. Okay, so next is the influence of pH. So most bacteria prefer to grow close to neutral pH. So these are neutrophiles or neutrophilic bacteria. Some can be acidophile, so they prefer to grow at low pH in acidic conditions. So once again, the real kind of champions at this are archaeobacteria, like Sophilobus and Ferroplasma. Now, some commonly used bacteria, Lactobacillus, is used for making cheese, yogurt, this kind of stuff. They ferment the lactose, acidify the conditions, and then most other bacteria can't grow, but the lactic acid bacteria can still carry on growing. That's very important for food conservation, of course. And then on the other side of the thing, then you have some kind of bacteria that prefer to grow at high pH. So think about some names here, Lactobacillus, if somebody asks you in an exam, give an example of an acidophilic bacterium. Lactobacillus, easy one to remember. For alkalophilic, this one's very easy to remember, right? Bacillus alkalophilus. Hey. So try and remember that one. So when bacteria are growing in acidic or alkaline conditions, the pH of the cytoplasm stays close to neutral. So that means that somehow these bacteria are kind of struggling against the pH outside the cell and trying to keep a neutral pH inside. So how does that work? So for E. coli and Salmonella, for example, if they're starting to grow in acid conditions, they'll express a proton ATPase, so they'll use ATP to export protons out of the cytoplasm. So they'll try and, actively use energy to keep the pH neutral inside the cell. Now, some kind of extreme of alkaline loving bacteria, what they can do is exchange sodium ions for protons. So they exchange sodium inside the cell for protons that are imported. And they try and maintain a slightly more acidic pH in the cytosol compared to the alkaline conditions outside. Okay, but in both of these cases, the bacteria have got to expend energy to maintain close to neutral conditions inside the cell. Now, next thing we have to think about is salt. Okay, so we're going to run out of time in 10 minutes. It's not good. Well, come back on Thursday, I guess. Once again, most bacteria prefer to grow at, 0.1, 0.2 molar salt. Many can grow down to, almost very, very little salt in the external medium. You've got to have some. And they won't grow very much at all if salt is higher than 0.5 or 1 molar. Now, I've looked for a word to describe these bacteria. Okay, so for temperature, you've got mesophilic bacteria, bacteria that like to grow between 20 and 40 degrees. For pH, neutrophilic bacteria. But for bacteria that like to grow with kind of normal salt conditions, I didn't really find a word in the biochemical textbook or online. So I'm just calling them non-halophiles, all right? And then after that, you've got halotolerant, halophilic, and extreme halophiles, all right? So halotolerant, they can grow up to 3 molar salt outside the cell, but they still actually prefer quite low salt conditions. So Staphylococcus are like that, okay? Staphylococcus aureus, it's halotolerant rather than halophilic. Okay, how can you tell the difference? Well, you can try and culture them at 2 molar salt and 0.2 molar salt, and you'll see whether they grow best, okay? They'll actually grow better in lower salt conditions. Then you've got actual halophilic bacteria, and they require high salt conditions in order to grow. So just trying to look for an example of this. This is ectothiorhodospira. It's a purple, non-sulfurous bacterium, I think, photosynthetic bacterium. It grows in kind of salt flats, like les marais salants, where you produce salt from seawater. Then the salt concentration can become very, very high in these pools of salt. Most bacteria can't grow. So you also have some halophiles. There's also not very much to eat in there, okay? So you get halophilic photosynthetic bacteria growing in there. And then really the champion of this is another kind of extremophile archaeobacteria, halobacterium, which grows like in the Dead Sea. Very, very high salt environment. Okay, so how do bacteria react to extreme salt conditions? Too much salt or not enough salt outside the cell. So that's what I mean, okay? Not enough salt, you'll have a hypotonic exterior. Too much salt, the exterior will be hypertonic. So let's think about this one first. So with a hypertonic exterior, what the problem is that all water inside the bacterial cell will tend to diffuse out, and everything in the bacterium will become totally dehydrated. So they've got to try and retain water by increasing the osmotic strength inside the cell. And the way they do this is to accumulate or build up a high intracellular concentration of what are called compatible solutes. So they can be like amino acids. So they're in solution, they have a charge, and so they increase the osmotic pressure inside the cell, but they don't really interfere with the function of enzymes or the pH. Okay. One of them, which is very frequent, are these glycine betaine molecules. So this is the N-terminus. This is glycine as an amino acid, but instead of having 2 or 3 hydrogens here, you've got methyl groups. So this is glycine betaine, so it's very frequently used as a compatible solute. So most eubacteria will do that, and in extreme halophiles, they can accumulate potassium inside the cell to compensate for the excess of sodium outside. And the potassium ions will keep the water inside the cell. Of course, this isn't really, this is an extreme adaptation to the conditions where these bacteria live, and they don't grow in what you would call normal conditions. It's basically not normal to have a way high concentration of potassium inside the cell. Okay, so if bacteria are growing in a hypertonic medium, the problem is the opposite. That is that water would tend to diffuse inside the bacterium because, you know, you've got concentrated salt, protein and everything inside there. And this will increase osmotic pressure inside the bacterium and put stress on the cell wall. So one of the things they can do is they can polymerize any kind of small organic molecules that they have into inclusion bodies. So it's almost like the opposite of releasing compatible, or, you know, accumulating compatible solutes inside the cytosol. This time they're all going to be concentrated in the inclusion body, so they don't contribute to the osmotic pressure inside the bacterium. So, you know, they'll all polymerize and they won't count so much for, coordinating water molecules. And the other thing they can do is also just, you know, release some into the extracellular space. And bacteria have mechanosensitive pores in the membrane. So if the osmotic pressure is too high, okay, so water is entering the bacterium here, the pressure inside the cell is too high, mechanosensitive pores in the membrane will open up and then solutes can come out. And then, you know, you won't have so much water diffusing into the bacterium. Okay, I'm going to just go through this because it's a very related concept, the activity of water. Okay, so salinity is kind of one particular case of the medium where water availability will be higher or lower according to the salinity, and this will impact on bacterial growth. And that's also true when bacteria are growing in a more complex substrate, like, I don't know, some porridge or a bowl of mayonnaise or some minced beef that you left in the fridge. Okay, so these are complex substrates, or a cake, something like that. You get it? And all of these like complex media will have some water in them, right? Okay, they've got some water content, but the water inside these different foodstuffs is not going to be particularly available because a lot of the water molecules are going to be interacting with whatever biomolecules are in there. Now, this idea about the availability of water is, whether it's available to take part in a chemical reaction. And that's also true for whether it's available to be used by bacteria. Now, I don't want to go into the physical chemistry here, but basically the way to measure this is something called the activity of water, which is a quantity that can be measured for different liquids and also different solid foodstuffs. Okay? And the basic idea is, the less the water is available here inside the foodstuff, solid or liquid, the less it can really evaporate out of whatever it is you have. And so if all your water is available, it can all evaporate, and it will give you, 100% relative humidity in the atmosphere above whatever it is. But if a lot of the water is interacting with, carbohydrates, fatty acid, protein molecules, then it won't be able to evaporate very much, and the relative humidity that you'll observe over the top of this plate of spaghetti or whatever will be lower than what you get with pure water. So basically, the activity of water is 1/100th of the relative humidity which is released by the substrate, whatever it is. Okay, bowl of milk, some cake, a fish. So you can apply this to any kind of food stuff. So it's very important in food microbiology. So I'm just going to ask this question. We'll do this and then we'll come back on Thursday to carry on, okay? Just to see if you've understood something about the activity of water. Now let me open the activity. Wait. Activity of water. Okay, here we go. Okay, we're kind of run out of time, so I'll just very quickly go through to the answers here. Okay, yeah, good voting, fast voting, very good. Okay, so the scale goes from 0 to 1. Yeah, that's correct, right? So 100% relative humidity divided by 100, that gives you 1. That's the maximum you can get. It's equivalent to the water content of a substance. That's not true. Okay, two different foodstuffs can have the same water content in terms of, percentage or the amount, but depending on what other molecules are in there, the activity of water will be will not necessarily be the same. So this is very important to realize. This is not actually the same thing. Okay, because depending on how much the water molecules interact with everything else you've got in there, they'll be more or less retained. Okay, so this is not true. 2 is not true. 3, very surprised to see this. Think about it. If your salt concentration goes up, the water that you have is more or less available. Less available, right? Because there's going to be some of it getting around the sodium and the chloride ions. So in fact, this is the opposite. In a highly concentrated saline solution, the activity of water goes down because it's not available. Okay, so this is the other way around. And finally, 4 is absolutely correct. Okay, so see you all on Thursday and think about the VBNC.